Hi, everybody. Welcome to the Listen Town Hall. Actually, I'm on a train right now, so let me just tell you, this is under unique circumstances, but everyone else is in secure locations where they'll be able to help in case I bump off. But one, we wanted to really highlight how special this is. We're so thrilled to be able to have a meeting with people who are participating in the study. We're so appreciative of everyone's time and effort and the ways that they've been able to contribute, the messages that they've sent us, the information that's being provided. And we can only do this through the approach that we're taking, which is that each of you is actually a member of the team. And we feel like we're just beginning this. I mean, we're just in a point where we're able to invite people and to start talking about the plans that we have. We've just reached 1,000 people. The group includes people reporting long COVID, people reporting vaccine injury, and people reporting both. Let me just say, we're just starting to reach enough people to start producing insights from the surveys and medical records. We encourage all the Listen participants to complete all the surveys. The more information that people complete, the more that we can use it together, and to connect all the records. Without all those connections and without the surveys, then we really don't have anything. So we've got to be able to pull together as much information together as we can. And we also appreciate if you can spread the word. We're interested in people with long COVID, with vaccine injury, and also those who have neither, because ultimately we're going to need a control population, people who are not suffering from either of these things that we can compare to those who are, to try to make useful scientific inferences. And we're also working hard, not just about describing this condition, but seeing whether or not we can test strategies that can help. And we hope that within a couple of weeks, we'll be able to announce the first clinical trial that we're going to be trying. And we look forward to communicating you about that and getting people in line and ready to participate, and to be able to work with all of you in a way that enables people to be part of it. But I think the most important thing that we want to communicate is gratitude and thanks for being part of this. And in the course of this, we also recognize that you guys have joined on a leap of faith. You've provided, many of you have already provided information. You've given us information that we hope to be able to turn into knowledge. We want to work with you to hear your questions, for you to hear what our plans are, for us to work together as partners, and to be able to make progress together. So we know that this has in some ways been frustrating in the sense that it's been started, but we haven't had any real meaningful findings yet, but we're just getting on our way. So I'm not going to tell anyone to have patience with us. You should always be impatient with us, but we want to work with you as close as we can to help make progress. With that, let me turn, and I apologize for being on the train. Actually, it was unanticipated. So thank you for sticking with me on that. And let me hand it over to Akiko for some remarks from her.
Yeah, thank you, Harlan.
And just to say, everyone, I'm Harlan Krumholtz. I didn't even say. I'm Harlan Krumholtz, and let me introduce Akiko. It was time.
Thank you, Harlan. And thank you for your dedication in joining us from a train. Really appreciate it. And thank you to all of you from Listen for joining us. Delighted to be with you again. I've really enjoyed interacting with you so far with the last town hall meeting, and Bornali and I did a follow-up because we had so many questions that we couldn't answer during the meeting. So there is a video up on the Kindred site that if you're interested, hopefully we addressed all the questions, but there may be others that you're welcome to submit to on the chat or question today, and we'll leave plenty of time at the last half of this meeting to have Q&A sessions. So please do ask us questions. Just like what Harlan said, we're very, very excited to be able to work with all of you. As I sort of presented last two times, I guess, there's a lot of work going on. Some of the work that I shared with you from the Mount Sinai Yale long COVID research, but also we're going to be expanding and extending those kinds of research to, with the Yale Listen study, to not only long COVID, but for vaccine adverse events and people who had a combination of long COVID and vaccine adverse events. And one of the emphasis that I made last time was the remarkable similarities in the types of symptoms that people are experiencing as a result of those three different conditions, as well as the pre-existing conditions and pre-existing symptoms that people have had in this 3 cohort. One of the things that we would love to do is to, so that was Bornali's analysis, by the way. One of the things that we are really excited to do is to be able to include control groups. So right now we have great participation from people with these 3 different conditions, but we do need people who are who don't have these conditions in order to compare and contrast the types of symptoms that people are experiencing or pre-existing conditions that people may have, and ultimately to compare the immune profiles. So we encourage people to participate, even if you, especially, if you don't have these types of conditions, but you're interested in contributing to research, this is a very important thing to do. The other thing that we would love to do in Listen is to be able to collect symptom data from people with these 3 different conditions. So, I know that the Kindred Listen team has already distributed the questionnaire for the symptoms, but we'd love to be able to get into that data as well. And ultimately, we will be asking some of you to join biological research analysis, looking at immune phenotyping by collection in saliva, and we will compare that to the different symptoms. So just to kind of recap what we discussed last 2 times, essentially we're finding immunological features that are clearly associated with long COVID, such as increase in exhausted T cells, increase in activated B cells, reduction in circulating central memory T cells, as well as reduced levels of cortisol. And that is the most uniformly and strongly correlated differences that we see in the long COVID. And then I also touched on the sex differences in symptoms and immune responses. That's a paper that we are currently working on to try to understand what symptoms are differentially felt, experienced by people of different sexes, as well as immune phenotype that are associated with those symptoms. So those are some of the ongoing research. Some things that that we are starting to do, but it's very, very new, are the microclot analysis and platelet activation phenotyping analysis. This is in collaboration with Professor Rezia Pretorius, who kindly came and visited us at Yale to show us how to do these analyses. And even though we only collected a few samples to do the analyses, we are seeing significant activation of platelets from people with long COVID. We haven't looked into the vaccine adverse event samples yet, but these are some of the new types of analyses that we'll be doing. And I know that the Professor Pretorius's group has reported significantly elevated activation of platelets and microclots in people with long COVID. So that's something that we'd love to be including in the future analyses. So yeah, these are some of the current and new thinking that we have and are planning to do with you all in the Listen study. And we'd love for you to be able to participate, if you can and if you're willing, for the biological analyses as well. And we'd love to be able to share with you some of the findings from these types of analyses. And of course, as with all research, we need to be able to do the analyses in sufficient number first to be able to compare on these immune features. But once we have enough number and once we have confidence, we will be sharing those data back with you, and we'll be thinking about it together. So that's very, very exciting for us. Yeah, so, and I'd be happy to answer any questions that you might have in the last half of this program, but I'm going to turn it over to Bornali to talk about what other measurements we're doing for the immune phenotyping.
Thank you, Akiko. So I'll start by sharing my slides. I hope everybody is able to see it. So, so we have been receiving a lot of questions about, what kind of surveys you are taking, or also about biospecimen collection. So I just wanted to begin with the study design. So for everybody, this is important to know that this study is divided into two parts. The first part of the study is analysis of the surveys that you take, the kindred surveys that you have been taking, and also the medical records, which makes it very important that you also connect your medical records so that we can analyze your data in an unbiased manner to get correct results from our analysis. So this is the first part of the study. This part of the study, every participant from any part of the world can join this study and take the surveys and participate in the listen study. So this is the first part of the study. Based on the analysis of the surveys, as well as going through your medical records, which we cross-check many a times, we will initially select subgroups of participants for biospecimen collection. Those who donate blood and saliva to us, on the very same day, they are also asked to take a few more surveys, which helps us find out their current symptoms. And we also go through their medical records to see if they have taken any tests so that we can correlate the surveys with them. So this is the basic study design. The biospecimen collection portion is only including residents from mainland US right now with the funding that we have. And so this is how we have designed the study. So with this, I would next talk about immunophenotyping. And to begin with the very beginning, some basic descriptions. I know many of you know a lot more than what I have in the slide, but I just wanted to begin with the very basics. Sorry. So our immune system, when we talk about immunophenotyping, we are talking about interaction with the pathogen and the aftermath of it. So our immune system protects us from harmful substances and foreign microorganisms, including viruses, and comprise of different cell types. Monocytes mature to form macrophages, which can engulf and digest debris and invading microorganisms. Neutrophils are another kind of engulfing or phagocytic cells. Eosinophils can act on parasites. Other than that, we have the dendritic cells or DCs, which engulf microorganisms and present their protein fragments on their surface to activate T cells. And when we talk about T cells and B cells, the different subtypes that have been spoken about and written about in our publication as well, these T cells and B cells circulate within the body, and when they encounter foreign bodies or microorganisms, T cells develop into activated T cells, which Akiko spoke about, with many different functions involved in protection. T cells are of two types, namely CD4 and CD8 protein-bearing types. B cells, upon activation, differentiate to produce antibodies, or are called antibody-secreting cells, as well as they generate memory B cells. Our data has also touched upon double negative B cells, which have been associated with inflammation and autoimmune diseases. And then we have the messengers or cytokines. Cytokines are small proteins that cells use to communicate with each other inside of our body. They quickly transmit messages to different parts of our body, helping to moderate the behavior of different tissues and systems like the immune system and the nervous system. The cytokines may be mentioned here or in any of the reports that we will provide to you as interferon gamma, TNF alpha, IL-6. These are just examples, one or two examples of that. Now moving further to study design, what I wanted to show you is what we are going to study. So of course, you have already understood that we need your surveys. We need your survey responses to understand the disease better. Right? We need those surveys because we are going to analyze the surveys. We are going to look into your medical records, which makes it imperative that we have that medical record connected to the Kindred or the Listen platform. Next, what are the different experiments that we are going to do? So, of course, we are going to look at the TCRs that we have so as to understand what are the pathogens against which they have been generated. That's one. Next, there will be multiple ELISAs that we are going to do. So what does ELISA do, or what question are we going to ask from here? We will be looking for autoantibodies. We will be looking for antibodies against multiple analytes, such as different kinds of viruses, bacteria, what kind of infections you have had in the past. We will look at it in multiple ways, which include rapid extracellular antigen profiling, REAP, as well as serum epitope repertoire analysis, which can tell us what pathogens you have encountered in the past, as well as at the present. And at the same time, we'll also look at autoimmune antibodies. Looking at viral antibodies is another thing that we will do. So SARS-CoV-2 antibodies will be looked at for different parts of the S protein as well as other proteins. And then we will also look at the efficacy at neutralization, which these antibodies have. So that's another experiment that we are going to do. At the same time, as I have spoken about the different kinds, so I have spoken about the different kinds of cells. So we also have a technique by which we will be able to look at what are the different subsets of cells. So how do we do it? The different cells I spoke about, actually, these cells have different kinds of proteins sitting on their surface. On the basis of antibodies, which can recognize those, we will be able to separate out different types of cells and calculate the numbers and give you a fraction calculated number of each of these cell types by doing a technique, using a technique called flow cytometry. So other than that, as Akiko has mentioned, we are very much interested in platelet activation, and we will look at microclots and platelet activation using all the assays that have been designed by the different scientists that she has already spoken about. So this is what we are going to do. And I know all of you have lots and lots of questions regarding how to connect our medical records, or which surveys have I taken, why haven't I received a survey? And we talk a lot about it. We exchange emails about it. So we thought there are two very talented people who are behind all the processes. They are both working for Hugo. One is Tanya, who is the product manager, and the other person is Emma, who's the data engineer at Hugo. And we have both of them today to demonstrate how it should be done and also respond to all your questions. So keep your questions ready. And I think I will stop sharing my slides and I will hand it over to Tanya so that she can walk with you through the process of connectivity issues, everything. Thank you, Tanya.
Thank you, Bernali. I'm going to go ahead and share my screen. Hello, everyone. All right, give me one moment. Can everyone see my screen? Perfect. All right, so this is nothing that you're unfamiliar with, but this is our Kindred platform. And I wanted to just highlight a couple of items that may be confusing or people have questions around that I can definitely clear up for you. So you walk into the Kindred platform, and the first thing that you're going to see is your active tasks. This is what Bernali is referring to as the surveys that need to be completed and how important that is for those to be completed to analyze the data. So they open up right into the survey, into a new tab in your browser or on your mobile device. So you would open them up that way, and this is the structure of our surveys. The most important thing is to consistently complete the surveys, because when you complete a survey, it's then backed with another survey that is all based on the questions that you answered. So it's very, very imperative that you complete the surveys or any tasks that you have up in this task carousel. It really helps with the research and anything that we are trying to gather for all the data analysis. So I wanted to definitely emphasize this carousel and the accessibility of it. I'm going to move further down. This is our content feed. So if you are in the Listen Study in the COVID community, so you're going to get Listen Study articles that are extremely helpful. You can listen to any of our town halls. There's also Kindred contributors where they tell their, the vaccine injured or long COVID, they tell their stories. It's very informational, very interesting information that we post. So I encourage you to read those definitely as often as you can. They are updated quite often as well. All right, so one more item that I want you guys to just take a look at within the platform, which I'm not sure if you guys know is accessible to you, is this My Account button up here in the top right corner. So if you go to My Account, you are able to fill in your first and last name. When you enrolled, it did take the city, state, and zip. If it's not in there, you can add it in there. Phone number. date of birth, and your communication preference. So if you have your phone number, you can choose to have your communication sent SMS, email, or voice. I wanted to let you know that whatever works best for you and that you can customize that within this My Account page. What's also nice under this menu is if you have a question, you can contact support, and it will take you to our frequently asked questions, and it'll help you guide you through any problem or issue that you're encountering or any question that you may have. And we also have our privacy and terms of service. So I wanted to highlight the My Account feature. We also, of course, you guys have the Listen Study, which you can click into, and it is only Listen Study related. And I'm sure that you guys are quite aware of that. But the biggest emphasis that I'd like to make is on data connections. So data connections, it is super important that we have this information of your medical records. It is the key contributor as to understanding the medical diseases, any current events that have happened since the last time that you've been seen. So I want to go ahead and go through a Q&A of anything that is confusing or about this process. So I'm going to go ahead and jump right into how to connect your records. Okay, so what you would do is you would go ahead and just click this Add My Data Connections. And then it says that Kindred uses Hugo Health to safely and secure your records. We connect with hundreds of providers. So not only healthcare, pharmacies, devices, applications, insurers, payers, etc. And that we only share your data with your permission. You can manage and revoke at any time. And I will show you how you can do that. I'm going to press continue. So what we do is we have a list of popular connections just to give you an idea of what kind of different clinical devices and pharmacies that we have to offer. But there's also this wonderful search field. So what I'm going to do is I'm going to go ahead and search for a provider in my area. I'm in the Bay Area, so I'm going to go ahead and search Kaiser. And you'll see that all the Kaiser locations pull up, and you can select which one that you are affiliated with. So let's say that I'm affiliated with Baldwin Park. I would go ahead and press the button to connect. I would enter my Kaiser Permanente Baldwin Park credentials into these fields. You would check off this box that you are authorizing the use and storage of the credentials and your health data through the Hugo Connect process. I want to emphasize this as well. Your credentials are securely stored and encrypted both in motion and at rest. With using a management system that is strictly controlled, credentials are never shared and are solely a part of your account. You can change or revoke access at any time. All right, so I'm going to go ahead and submit. And there's this fun confetti page that shows you that you have submitted and that record. You can go on to add another. So let's go ahead and do so. Let's go ahead and choose a pharmacy this time, and let's go with Walgreens. Same thing, you can go ahead and connect, put in your Walgreens credentials, and submit. Some of the pharmacies or providers or healthcare systems have an external question or an additional item that you need to, you have to redirect to a different page or an additional question. This one happens to with Walgreens, so I'm going to go ahead and submit an answer. And this is all test data, so this isn't anything real. Just want to show you what it looks like. When you are all done connecting your records, you can click this button. You can see what you've already connected. And if you click out of this box, it is now showing up under My Data Connections as connected. Now, let's say that you would like to that this says it needs attention. Maybe the password that you entered wasn't correct, or maybe you need to re-authenticate yourself. You can go ahead and select this wrench here and go to Repair Connection. That will bring this Baldwin Park Medical Center right back up so that you can re-enter your credentials and hopefully have a successful login. If you'd like to revoke access at any time, the same applies. You would disconnect. You would receive this page to unlink the connection, and it states that you're unlinking the connection, and this will prevent any of your connected services from accessing your data in the future, and you can unlink that way. And so I wanted to definitely show you all the different types of things that you can search for, any questions that you may have, and and go from there. Anything that I didn't cover that you feel needs to be covered?
So there's a question in the box. So when comes to adding medical records, every time I connect to facility portal, it will connect, but then connection will be lost. I also keep getting sent duplicates of surveys, that I have already filled out and finished. When I get email saying need to complete, if I have already completed, do I really need to fill out again?
If you're still receiving survey emails, then that means that there is a survey to complete still. So if you log into your Kindred platform, you should see a survey up in my active tasks if you are still receiving an email to complete a survey. So that means that it might be in progress, but not fully completed, because it will still show up in the carousel until it is fully completed. If it's in progress and you stop at a certain point, if you press the button, it will restart at the time that you stopped the survey. I hope that that helps.
Yeah, Tanya, there's another question that a couple of people have asked is, if none of the providers are linked, what is the process?
If none of the providers are linked, meaning you can't find a provider under?
I believe so, yeah.
Okay, so let's say you can't find the provider. So let's just search here. All right, so you can't find what you're looking for? Let us know here. And this is where you can go ahead and you can submit a request to us saying, hey, I can't find my provider, and that we will do our best to go ahead and locate that provider and try to get that added. Does that answer that question?
Perfect. Thank you.
You're welcome. So as you can see, this needs attention already. It updates rather quickly because I was using test credentials. So just to let you know why that says it needs attention. It's just the test credentials that I was using, but in a normal situation, it would be connected with the correct credentials entered. Any further questions that I am?
So here's one from Sheila. I see that. I signed up back in August for the survey, but have received nothing. Can someone help me get into the pipeline? So I think Talia has already sent out an email ID where you can ask all of these questions and get a response. Do you want to add any other suggestions?
I would need to have more information to see what exactly this person's encountering, but I would be happy to assist or stay after to figure out how that could be occurring. Yes, I want to hear and know about any possible, disconnect or any difficulties that anyone's having within the system other than finding their connection, because there is a way to submit that and we can try to obtain that. But yes, I can definitely follow up on that.
So I also see that somebody is asking, Angie here is asking, how often should we check the app for surveys?
You should be receiving emails for any new surveys that are assigned. So I would, it's always a great thing to visit the app, a couple of times a week or as often as you'd like to see if you have any active tasks. But we will send email correspondence when there is a task that needs to be completed, and that would show up. And whatever task we would send an email about would be up in this task carousel here. And there might even be one that says connect your health records. And what you would do is you would press on this arrow, and then it would pop up this screen to encourage you to enter your medical record information.
Thank you, Tanya. If there are any other questions, please type it. And I can see many are being answered in real time now.
Bernali, if I could just jump in real quick. And I wanted to explain a little bit about how our data collection works. A lot of the connections that we support are through an EMR provider called Epic. You'll notice that when you go to connect your credentials, a new tab will actually open and you will have to enter your credentials there. I'm seeing a question in the chat about if you have multiple Epic connections, do you need to sign up with each one? The answer is yes, because you are consenting on a sort of hospital basis to share your information and not the letter or the EMR basis to your information like that. And I just wanted to share how sort of new and novel this process actually is. We, well, not we, but if you guys are familiar with the Cures Act, which was passed in 2016, that basically said that your healthcare providers need to share this information when you request it. And things like that actually take a very long time to get going. And, you know, then COVID happened and things kept getting pushed back. And we've reached this state where the deadline for full compliance is actually coming up pretty soon. It's the end of December 2022. So a lot of these processes that you guys are going through and almost sort of testing out for us, they're works in progress. So if you do see anything weird, you encounter a connection that you think should be there that's not, or you're finding that, you know, you connect and you keep getting needs attention, we encourage you to reach out to kindred_support@hugo.health. The connection related issues actually tend to come straight to me, so I can get you going and make sure that we have everything working as smoothly as possible. I'm just going to check and see if there's any other questions that I can answer while I've got the floor here.
Thanks so much, Emma, for that explanation.
Yeah. I just tried reconnecting Needs Work account. How long does it take for a new connection to show up? So that's actually a great question because we get this one a lot in our support. It's not instantaneous. So the way it kind of works on the back end is once you finish, there's a message sent out to the rest of our system and it basically queues up a job. So it's like first in, first out type deal. So if there's a whole bunch of other people connecting at the same time, it may take up to, you know, 2-3 minutes for that new status to show up. So give it a little bit, maybe even refresh the screen in a minute or two. And if you're still seeing that needs attention, go ahead and reach out. How far back do you want my health records? So when you first give us permission to go and access your records, we will pull basically everything that you give us permission to as far back as your electronic health record actually stores. And this varies based on different providers. I know my personal stuff I could see as far back as I was born, but I was born in 1998. So some people might not have that same sort of longevity in terms of their records. And then what if our medical provider is a solo practitioner? Again, if you reach out to Kindred support@hugo.health, we can, you can send us your practitioner's info and we can see if we could get you guys connected.
Emma, there's a question I see. Yep. Are there HIPAA protections in place when sharing medical records?
This is also a really good question. So Hugo Health isn't what's called a covered entity under HIPAA protections, but we care greatly about your ability to share and protect your own health data. So we actually attest to and implement all of the HIPAA standards on our own, which, you know, as someone who actually uses the Kindred platform, it's great because it means that there's less vulnerabilities when we're transferring your information to different places. It means that when it's stored, there's actually even more protections in place. Your information is actually completely de-identified. So if there were a breach or something, which not usually the case, they wouldn't be able to link any of that back to you whatsoever. And we really do believe and attest to HIPAA policies. There's also something, in case anyone's curious, called the Karen Alliance, which is an additional set of principles that different healthcare providers and health and information technology providers can attest to that have even stricter policies regarding sharing of data and all sorts of protections around that sort of stuff.
Hey, Emma, can you hear me?
Yes.
Hi, this is Harlan.
Hey, Harlan.
People do ask us about the issue about data sharing and security, and when they say we're connecting our records, what are we doing and what's going to happen with them? And I do think it's important to know the way this works on your behalf. Hugo's going, you may have just said this because I just got bumped off. I'll come back and say it again. Hugo, on your behalf, is getting your data to deposit into your secure cloud-based account and grow your data assets. With your permission, that data goes into the study. With your permission, the data can be used in aggregate for us to create some summaries about what's going on from the Kindred community, but you will never be identified individually. Your data is not going to be sold or moved or monetized. It's about empowering you for your data to come in and for you to give permission for your data to be used in this way. If you change your mind, if you decide you don't want to do this, you can. It's your data. Hugo does not own your data. You own your data. And we're trying to create the environment, the ecosystem, where you can put your data to work, but we're also going to fight like heck to protect your data and protect your rights and help you to get your data. Again, it comes in the way we conceptualize it. It's like we say B to C to R. It's like from the businesses or organizations that hold your data to C, to the consumer, to you. And then you're giving permission to go use it for R, for the research. And then when it sits within the Hugo community, we're not doing any funny business or have anything like, hey, because you're on this platform, we can de-identify and move it around. It's yours. And so what we're working on is a permission-based system, a sense of community, put this together, and try to use the platform for the purposes of trying to empower people to be able to do things with their data. There has to be a business model with Hugo. I mean, we want it to be sustainable. So in the transactions and partnerships, we're trying to make it so that there's enough revenue generated to be able to help support the platform to do this. But what we won't do is compromise on the idea that it's your data or do anything with your data in order to generate revenue. That's not in the model. The model is working in partnership with you for things that you, if you want to be part of Listen, that's great. We want you to be part of Listen. That data then can be shared with the Listen study. I wear two hats in that way. I'm on the Hugo side to try to help build the platform. I'm working with Akiko on the Listen side to try to help learn something with you about the conditions that you guys are facing and are challenging. And our intent purpose is to move as fast as we can, learn from you, and to try to make this into something meaningful for the community, not just our community, but the world community who are facing the same challenges. But I wanted to just say that because from time to time I see someone raise that issue, and we just want to say it really clearly and loudly that the data is always yours, and we're trying to, everything is going to be based on permissions, and that's just the way we'll work.
And there was one exciting piece that I wanted to show, if you don't mind. There was one more thing that is in the works that is coming very, very soon. And hopefully you can see my screen here. Give me 1 second. All right. So with the Hugo Connect feature here, what we're going to have coming soon is the ability to upload your records. So let me just move this bar real quick. So you're going to be able, these are the mock-ups for it. So you're going to be able to have an upload feature. So if you can't find your provider, or if you just receive something from the doctor, or if you have notes that are, no longer in your file, or what have you, whatever you'd like to send to be a part of your record, we can upload that file. So it would be as simple as finding this within Hugo Connect, and then just simply uploading, and then adding a name to the file. We're going to have some categories of what type of file this is, whether this is an X-ray or your medical records, immunization records, etc. And then you'll be able to date the form and continue. So I think that's another way to really try to get your documents and your information to us to be able to use that for research. So I thought that was an exciting way to kind of bypass not finding that provider at that moment. You can upload documentation as well. And that's what I had to share.
Thank you, Tanya. So I believe everybody was able to get some answers to your questions regarding connection of records and also about how we protect the data, and it's your data that remains there. I think next we will move on to listen to Cesar and his conversation about more diversity in the population that we have or the community that we have, and many other aspects which are also important for the study. So Cesar, it's over to you.
Thank you, Ornali. Hi, everyone. My name is Cesar Caravaggio. I'm one of the research coordinators of the study, and I wanted to speak with you on a couple of things. First, regarding diversity, we want to make sure that our group of participants is as diverse as possible because we want to be sure that we're capturing the experience of everyone, or that most people are experiencing in the United States or wherever you are. So we are making efforts to improve the diversity of our participants. And I wanted to ask you if you have any ideas on what can we do better to improve the engagement of communities that have typically been underrepresented in clinical studies. We're open to it. We're open to feedback. If there is something that you as participant feel that may be a barrier for other people to join the study, we welcome every comment that you may have. We want to have a rich and diverse population of participants. And so the idea behind that is that we can produce information that everyone can apply to themselves. So yeah, that's one point. If you have any ideas, any feedback regarding our efforts, they are very much well received. The second point that I wanted to ask you as participants is that in some of the surveys, particularly in the symptom surveys or in the diagnosis surveys, we found that a lot of you are clicking none of the above, meaning that of the list of symptoms or diagnoses that we're listing, you are not identifying the ones that you are experiencing. So I wanted to ask you too, if you think that we are keeping out some key aspects of your experience, please let us know. You can type in the chat or you can email us to Yale Listen Study, Listen Study at yale.edu, and we'll be happy to modify what we're doing to better capture your experience. I will type in the email in the chat so you can have it. And that's it. Thank you. I think now we can, yeah, Bernali, go ahead, sorry.
No, Cesar, are we taking questions now? Leave it to you.
Yeah, we'll be more than happy we start answering some questions that people may have. We can go over the ones that are still open, or if you have any at this point, please feel free to type it.
Yeah, so what I have is a list of 10, and we can have a few more maybe from the list which are still open. I believe Akiko and Harlan are both here, and we can start. Yeah, perfect. So we can start with a few questions, and then Cesar, you can add a few more from the open questions.
Sounds good.
So I'll start with the 10 that I have on my list. Akiko, there are a few for you, which I will start with. So the first question that we have here, will there be any efforts towards correlating immune features with symptoms? Example, distinguishing patients presenting with dysautonomia versus not?
Yes, absolutely. That is our ultimate goal, is to be able to link the symptoms to the immune features. We've already started to do some of that with the My Long COVID study and found, for instance, select number of people who have autoantibodies to certain type of G protein coupled receptors and sodium ion channels were associated with the tinnitus. So there is some sort of features that are coming up to be associated with specific symptoms, but we also want to do the reverse, as the questioner is suggesting, which is to take a particular symptom or syndrome and then go back and look at the immune features to see if there's anything unique that's coming up. So that's ultimately the goal.
Thank you, Akiko. So the next question is, Can you speak to whether in the initial findings you have seen dysautonomia in many long COVID and vaccine-injured folks? It seems to be common.
Yes, dysautonomia is definitely happening. I mean, we haven't studied the vaccine injured people yet, but we are hoping to do that with the Yale Listen study. But certainly with the mild long COVID, there are people with dysautonomia, and Dr. David Petrino, who really specializes in diagnosis and treatment of various symptoms, including dysautonomia, is our partner in this. So he's also a partner in the Yale Listen study as well. So we're very excited and fortunate to have an expert like him on board to help us with it.
Yeah, and I'll just say quickly that just as Akiko said, that the hope is to be able to provide almost like an atlas, a map of the different kinds of manifestations that people are facing, see who's similar and who's different, and then see whether or not we can reflect this in the immune, what we're calling immune signatures, the unique ways that the immune system seems to be signaling us around the different features of the immune system. And so just what you're asking is exactly what we're hoping. Can we get large numbers of people with similar clusters of symptoms and then see whether they have distinctive biological signals, which can serve both for diagnostics and therapeutics, help us be able to identify, monitor, and then ultimately find targets to treat for each of these people, because I think both of us, Akiko, believe that this is more than one disease. It's actually different people are being affected in very different ways. And that's why they're manifesting with very different symptom complexes. And it's up to us to sort of find the clues, crack the case, understand what it is that's causing this vast spectrum of symptoms. And for different clusters of people who are experiencing similar ones, can we find common mechanisms? And so that's really the hope. And that's why we need large numbers of people, because we want to, I think of it like an atlas. And it's almost like who lives in your neighborhood. This is an unfortunate neighborhood because everybody's afflicted by a sort of similar cluster of symptoms. But if we can get large enough people together, as opposed to what's happening now, where somebody goes to the doctor and people say, I've never seen this before, or I don't know what this is, that we start getting large numbers of people who are saying like, yeah, I'm not alone. There are a lot of people like me. There are a lot of people like me in the world. And if we can start bringing those people together and looking whether they have similar biological signals that help us understand mechanism and lead us to produce diagnostic and therapeutic advances, that would be wonderful. And that's the hope with this.
Thank you, Harlan. There's another question, which I will just present. So here is a person who's suffering from vaccine adverse events. And this person is asking, will you be contacting us to participate in the biological sample collection, or should we let you know we are interested?
Absolutely. So as I mentioned, we're very excited to expand our studies to vaccine injured people. And Bornali, Cesar, Harlan, and I meet every week to discuss the type of participants that we would like to recruit first. We don't have the resources to recruit everybody. We'd love to, but we don't have the kind of resource needed to analyze everybody. But we will be starting with a small subset of people and then expanding that as we hopefully raise enough funds to be able to look at immune features. But definitely our first set of vaccine injured samples will be coming shortly, right, Bornali? Okay, great.
And by the way, I think this is a really good idea that we could talk about, which is, do people want to express interest in being part of the biological part of the study? Then we'll have a long list of people who've already expressed interest. Like Akiko said, by the way, we're constantly looking for resources to be able to expand and do more of this. But, I think it might be a good idea, based on this comment, to think that we can create a waiting list of people who are interested. And that way we know that, these are people who are really eager to participate in this part. So that's a really good comment. Thank you.
Yeah, it would be great, Harlan, if Kindred, so the Listen site can have some sort of marking to say that if people are interested in donating their blood and saliva, it would be easy for us to just sort of cross-check that with the participants that we want to recruit. So maybe it's something that you can work on for us.
Thank you, Akiko and Harlan. The next question is about the recent MDPI article, which has shown that persistent spike protein in brain and heart of a patient who died 3 weeks after third vaccine. How will you be assessing for persistent spike protein that may be triggering an immune response?
Yeah, that's an excellent question. We'd love to do tissue level analyses, but obviously taking biopsies from brain and lung and places that are difficult would not be possible. We are, so the autopsy studies and other studies are coming out where people are starting to look at these issues. But even with the COVID samples, this has sort of been a difficult thing. With the Recover, though, they are having an entire sort of, funding for a collection of autopsy samples from people who've had long COVID. I don't think they are including vaccine injured people in that group, but aside from having a tissue sample, which is maybe difficult in some cases, but there are some efforts to look at circulating spike and circulating antigens, at least that's easier to collect. And we are actively working on looking at circulating spike from people with long COVID and people with vaccine injury. So that's just a proxy for something that may be happening elsewhere, but that's sort of what we are, immediately planning to do.
Thank you, Akiko. The next question is about a recent Swedish study found antibodies for latent viruses in saliva, but not always in the blood. Will saliva be tested for reactivated viruses as well in case it's missed in the blood?
Absolutely. And that's why we are including blood collection in the LISTEN study. So with the My Long COVID study with Mount Sinai, we didn't have access to the saliva and we really wish we did. But in the Yale LISTEN study, we will be asking participants to donate their saliva and blood so we can look at both mucosal and systemic immune responses. And in fact, we are planning to look at viral genome as well as antibody reactive to virus antigens, both in saliva and in circulation.
So the next question is about autoantibodies. So you have mentioned not seeing elevated GPCR autoantibodies in those of long haulers compared to controls. Can you comment on that, please?
Right. So as I shared with you last time, we are collaborating with Dr. Aaron Ring's laboratory here at Yale, who developed a technology called Rapid Extracellular Antigen Profiling, or REAP. And according to that analysis, which looks at over 6,000 different antigens that are expressed on the surface or are secreted from human cells, we do not see a consistent. First of all, we don't see a public or universal antigen that people are reacting to. So it's not like you have a specific antigen that everyone is responding. That's definitely not the case. We also don't see elevated levels of autoantibodies against these 6,000 plus autoantigens in long COVID patients versus the controls. So there's no unique pattern. There's no increase in autoantibody reactivity and intensity. There are, of course, various different autoantibodies that all of us carry. That doesn't mean that we are going to have disease. It's just something that B cells make, but it's just sort of quiescent autoantibody. And that we are seeing in healthy control, convalescent control, and long COVID. But that doesn't mean that there doesn't exist autoantibody that actually causes disease in a subset of people. So I already mentioned about the sodium ion channel, but there may be other autoantibodies against intracellular antigens or nuclear antigens, who knows, that could be causing disease, and that we're not dismissing those. About the GPCR, yeah, there are many different ways of assaying autoantibody against GPCRs, and depending on the sensitivity and specificity and having the right kind of control groups to compare, that's what I was emphasizing last time. And if anyone has studies that have included the proper controls, please do send it to me. I may have missed that. But so far, I haven't seen great controlled studies to compare autoantibodies to GPCRs in various different conditions with the controls. And so that was sort of related to what I said last time.
Thank you, Akiko. I think I'll pass on to Cesar now so that he can ask a few more questions. Over to you, Cesar.
So I think it's almost time to be respectful of everyone. So I know that there are a lot of open questions. I'm sorry that we won't be able to cover them all. Maybe we can do something similar or put together a document after the town hall answering most of them. But yeah, I don't think we have time for more questions, Bernali.
Okay, so I think we can list the questions and maybe put them up, like the responses later. Yeah, sounds like a plan. If there is nothing else to respond to, we are already running short of time. I think we will thank all of you, the panelists and the participants who have joined us. Please ask questions, write to us. Please finish the surveys whenever you receive an email, and also connect your medical records. These are all very important for us. And we would love to have controls who did not have long COVID, who do not have vaccine adverse events to report, or neither together. And we need these controls in order to compare and contrast. Thank you all for your presence here today. Thank you.
Thank you so much. See you again soon.